Good morning, afternoon, or evening, everybody. I hope you are doing well and stay healthy. Today, I would like to welcome you to a new Meet the Expert webinar session brought to you by Lallemand Forward, which is the platform we have covering educational and on-farm services as well as industry support. In this occasion, we are going to talk about a very interesting and actual topic, microbiota, and some of the ways of analyzing it. The microbiome, as you know, assumes a vital role in digestion, absorption, and assimilation of feed, as well as restricts the growth of pathogenic strains. Due to all these interrelationships, the knowledge about microbes opens avenues for researching solutions to support the management of swine production and improve animal health and well-being and production sustainability. The study of gut microbial ecology has undergone a revolution in the last 15 years. And in order to bring some light about this topic, it is a great pleasure for me to invite my colleague Emmanuel Appert on stage. Emmanuel, good morning.
Good morning. Thank you for having invited me.
You're welcome. Just one small issue. We have an issue with Emmanuel webcam. So regretfully, you can only see me. That's a pity, but we cannot see Emmanuel on the screen. But anyway, we are going to, the most important is the content that she's going to share. Emmanuel is an agricultural engineer who holds a PhD in animal nutrition and physiology from INRAE from Rennes. After 3 years as a researcher and lecturer in the engineering school, she joined the industry as scientific affairs manager in animal nutrition for Tereos, and then as research program manager at Royal Canin, becoming also member of different regulatory, we have an issue here, of different regulatory or scientific associations like ILSI Europe, FEFANA, Animal Nutrition Commission of the EAAP, and more. In January 2019, Emmanuel joined Lallemand Animal Nutrition as the Global Manager of the Centers of Excellence. Her central mission is to support the scientific excellence of the team and ensuring the research activity is aligned with the company strategy. The structure of the webinar is going to be as follows. We will start with a presentation by Emmanuel, which is actually the central part of the webinar, and whose objective is to tell us in simple words how we can analyze and interpret microbial data, also including some correlations with the outcomes we can extract in swine. After Emmanuel's presentation, I will take 5 minutes of your time, I hope it is not more than that, to show you some of the research we have done with our specific live yeast probiotic, Lebus LSB, and after that we will dedicate some time to answer questions from your side. On that respect, you can use the panel that appears on the right-hand side of your screen, and apart from downloading the handout of the webinar, you can open the question section and write your specific question. Emmanuel, before going to your presentation, I would like to ask you about the concept of centers of excellence. What does that mean?
Thank you for this question, David. In fact, the Center of Excellence allow developing scientific excellence in Lallemand Animal Nutrition, and it is a unique and original link between academic scientific research, applied research, and the economic world. It will give us some preferred access to different analytical platforms, and we are in close collaboration with academic researchers, and it allows us to have a continuous improvement in our methods, especially.
Okay, thank you very much. And Emmanuel, could you all tell us more about what is the structure? How are those centers of excellence structured?
Yeah, in fact, we have 3 main centers of excellence. The first one is the Forage Center of Excellence. The other one is a Ruminant Center of Excellence, and the last one is Monogastric Center of Excellence. Each center of excellence is located inside academic research center, and again, they have each researcher of this center of excellence have access to last techniques, last field research, and so on. So it's really an advantage for us to advance in our research and in our reflection.
Okay. Thank you very much, Emmanuel. So I think now we can proceed with your presentation. Emmanuel, the floor is yours.
Thanks a lot, David. Hello, everybody. It's a pleasure for me. Okay. I have still an issue with my PowerPoint. Okay, I hope you see the slide now. It's a pleasure for me to present to you the study of the microbiota compositions through amplicon sequencing. How to do it, where are we, and what are the perspectives? This presentation has been prepared in collaboration with my colleague Anna Rodiles and Carolina Schar. And just as a quick explanation of the composition of the team, in fact, we are a high-skilled team of 5 scientists, 2 project leaders, and 1 technician. And as I mentioned at the beginning, the Center of Excellence are dedicated to work on microbiota, and it is a unique and original link between academic research, applied research, and economic world. And we have preferred access to different analytical platforms. I put here 2 examples, Genotool, Get, and Cribion, which are platforms allowing us to do 16S analysis, but also metagenomics, metatranscriptomics, and metabolomics. We have 2 scientists dedicated to amplicon sequencing and omics analysis. The 2 colleagues who support me in this presentation, Carolina Schar and Anna Rodiles. The objective now of the presentation, the first one is to explain to you why the research on microbiota is living such a strong revolution. The second one is to understand the place of the amplicon sequencing in the landscape of the different methods used to study the gut microbiota and microbiomes. And we will go a little bit into detail on the amplicon sequencing to describe basic and key parameters to understand the main result obtained with the amplicon sequencing. The idea here is really to give you some tips to understand the main result we can obtain with these techniques. And as a fourth objective, I wanted to give you some perspective and field of research allowed by the high throughput sequencing and omics methods. I will give you the main take-home message that I would like you to have at the end of this presentation. The first one is that method innovations allow to study not only microbiota, but also microbiomes. The second one is that amplicon sequencing allows studying gut microbiota composition and structure, and it is based on amplification method and gene marker discovery. The main feature you will obtain with amplicon sequencing are the number of species present, their relative numerical composition, and the bacterial diversity defined as alpha and beta diversity. And amplicon sequencing, thanks to dynamic evolution of bioinformatics, allow also to understand the relationships between microbes. We can go a little bit further than the 3 main features, just to try to understand the interconnection between all the microbes and the integration of data from different high throughput methods, but also from phenotypic parameters of the animals, will allow holistic approach to study host microbe, microbe, and microbe environment interactions. But we have to keep in mind that to be successful, the tools available must deceive hypotheses and concepts, and we do not have to be driven by the methods to be successful in this research. As an introduction, the gut microbiota is a complex and highly dynamic environment. It is 10 to the 14 bacteria, that populate the gut, and it represents from 500 to 1,000 bacterial species in the gastrointestinal tract, and more than 40,000 bacterial strains. And just to give you an idea on the importance of the global bacteria we can have in the human body, it's the weight is from 1 to 2 kilograms, and it is comparable to our brain weight. And the importance of the microbiota resulted in a new definition of the individual, the holobiont theory, meaning that we are in a connection with our microbiota, and we cannot explain our physiology or response without taking into account the microbiota. The gut microbiota, it is a strong trend for a hot topic in research, and this is this recent publication which studies the importance of the number of publications with the microbiota as keyword, and you can see that from 2010 there is an exponential increase of the number of publications. Why? Because the molecular biology and the high throughput sequencing have changed our life. So the explosion of the microbiota study is really related to the new techniques. And before going more into details on the techniques, I wanted just to give you some definition about microbiome, microbiota, and metagenome. Microbiome, first here, it deals with microorganisms and their gene living in a specific environment, and it refers to the microorganism and their genes. So here, microbiome will imply interaction between microorganisms and their environment, and notably with the host. And microbiome, or as well as microbiota, will gather fungi, protozoa, archaea, bacteria, algae, and viruses. Microbiota, it deals with microorganisms by type, living in a specific environment, and contrary to what we call the microbiome, the microbiota will mainly refer to the taxonomy of microorganisms present. So the who is there. And finally, the metagenome. The metagenome are the genes of microorganisms in a specific environment, and it refers to the collective function of microbial genes. As I mentioned at the beginning, there is a very strong relationship between the advance in microbiology and the method innovation and the scientific discovery. And you can see that here with the invention of the microscopy and the knowledge of the existence of microorganisms, so the discovery of microorganisms, the research was really linked to animal and human disease as a consequence of microbial infection. And the pathogenicity concept appeared in this period. After that, with the evolution of the techniques and with the apparition of the biomolecular concepts, we went through another concept like microbiology and ecology. So it means that there were interaction between microbes. So only a small proportion of microbes are associated with pathogenicity, and this is the emergence of the microbial ecology. And in the last years, now we know that in fact everything is in close relationship. It was the holobiont theory I mentioned at the beginning, and we are today in the One Health concept. And then today, we know that there is ubiquity of microbial communities in association with higher organisms. And here, the high throughput sequencing and omics methods play an important role to decipher all this interconnection. So molecular biology and high throughput sequencing method have changed and will change still our understanding of microbiota. But for sure, to deal with this metadata, we have to do some bioinformatics and biostatistics. If we now go more into details on these different techniques, in fact, they will allow you to answer different questions. From at the beginning, I mentioned the microscopy when you want to study cells. Microscopy will allow to decipher the microbial phenotype, the colonization pattern. And we can also do some culture and culturomic to do species characterization and cellular function. When you want to evaluate the microbial potential, you can also have a look on the DNA. And with the DNA, you can do amplicon sequencing to study the community composition and the microbial networks. And you can also do metagenomics to study the genes, the genomes, and the gene function. And if you want to evaluate the metabolic potential of this microbiota, you can study the RNA and you will do metatranscriptomics. So you will study the gene expression of the community. And if you want to go towards the microbial function, you can do either metaproteomics to study the protein expression, or finally the upper level metabolomics to study the metabolite production and the microbial product. And today, as explained by David, we wanted to give you some tips on amplicon sequencing, so the method that will allow us to understand the community composition and the microbial networks. Amplicon sequencing, it's an easy method to describe microbiota, and it's quite cheap today, so it allows to do a lot of studies, and it is again to analyze a microbial community, and here we will focus on the intestinal microbiota based on DNA sequencing. We will use only one specific ubiquitous gene that is targeted, and it is then based on a gene marker sequence. And one bacterial species will be represented by one specific DNA sequence called the amplicon. In terms of result, you will obtain the qualitative analysis with the bacterial species identification or taxonomic affiliation. You will obtain the relative quantification. It means the quantity of one specific sequence that is generated will be proportional to the relative abundance of the corresponding bacteria. And at the end, you will be able to understand the community structure through diversity indexed and to build some networks. If we go back to the importance of the gene marker, the most used marker for bacteria is the 16S rRNA gene, which is present in all bacteria and archaea. It is in fact a non-coding RNA containing 1,500 nucleotides. And the specificity is that some regions are highly variable, while some regions are highly conserved. And this is what you can see here with the percentage of sequence identity. And you can see that in gray, you have very variable, hypervariable region, V1, V2, V3, V4, V5, etc. While you have the orange part, which are the highly conserved part. And this highly conserved part will allow us to design primers or to use primers. And the Illumina sequencing will sequence part of hypervariable region that will allow to identify the bacteria. And there is, after that, large database available to make the identification. If we look at the typical workflow of the amplicon sequencing, you have here a schema. And you will do, of course, you have different steps of method, and each step is very important as you can obtain some bias. So for example, the collection of samples and the storage of sample, which is the first step, are really important to make sure that your sample will be of high quality. And the second step will be DNA extraction. And you will have different lysis protocol, different kit used, and this will make some difference from one study to another one. So this is something to take into account when you see some results from 16S analysis. And the step after will be the PCR amplification, and you will target one region, for example, the V3, V4 region of the 16S RNA gene. And again, depending on the region you will choose, you may have some different results from one study to another one. And the last step is the Illumina sequencing, so the sequencing of your sequence. After that, of course, you will obtain a huge library, and you will have to do some bioinformatic analysis to obtain what we call OTU, Operational Taxonomic Unit, or ASV. This is basically the same, amplicon sequence variant, just the method of obtaining this is are a little bit different from one type to another type. But basically, you will have either OTU here or ASV, and you will obtain what we call an abundance table, OTU abundance table, or ASV. For the remaining of the presentation, I will speak about OTU, but can be also ASV. This is really basically the same. And you will have after the taxonomic classification from the domain to the species. But you have to keep in mind that for the 16S, most of the time we go to the genus level, not to the species. So now we will see what do the results mean. As I mentioned in the take-home message, the important feature of a bacterial community that we will obtain using the amplicon sequencing are the number of OTUs present, their relative numerical composition, and the bacterial diversity. We will obtain the three following parameters: the percentage, so the relative abundance of each OTUs, the alpha diversity, and the beta diversity. Then we can correlate this with several factors like piglet age, diet effect, physiological stage, etc., to understand the role that the microbiota can play in this. And to illustrate these three main parameters, I will use practical results from one publication which aimed to study the mucosal and luminal microbiota across swine intestinal tract. First, if we go to explain the relative abundance, the definition, in fact, it refers to how common or rare a species is relatively to other species in a defined community. It is the percent composition of an organism of a particular kind relatively to the total number of organisms in this area. So the objective when you want to have the relative abundance of your OTU is to see how many operational taxonomic units are differentially represented when comparing samples. And you will define your OTU and ASV at different level of resolution. I will explain a little bit. You have here a very practical and simple example. You have three samples here with different types of bacteria, and you will have an absolute number of the pink bacteria, the green bacteria, and the yellow one. So for example, in the sample one, you have one yellow, 3 pink, and 3 green. For the sample 2, you will do the same. And after that, in the second step, you will normalize your data, and so you will have the relative abundance for each sample in percentage. And practically, you will have a graphical representation, and you can see that OTUs will be gathered at different taxon level, depending on what you want to obtain. And the example here deals with the phylum level. So you can see from the publication I mentioned before, you have the ileum, the microbiota at the ileum, cecum, colon level in the lumen, and the same at the mucosa level, and you will have differential composition in terms of different phyla like Firmicutes, Bacteroidetes, and so on. And after that, once you have this proportion of phyla expressed as percentage, you will be able to do differential analysis using non-parametric tests. Now, if we go to the alpha diversity, the definition is the average diversity of a community within a sample. And here, the objective is to describe microbiota composition profile in terms of diversity and homogeneity. So there are two important notions in this alpha diversity. The first one is the richness, and the second one is the evenness. The richness is defined as the number of different kinds of microorganisms, while the evenness is the uniformity of the population size of each of the species present. In other words, you will use the relative abundance of the OTUs. So for the richness evaluation, there are different indexes that will allow you to estimate it. I put just few examples, but there are many. The KO1 and the number of OTUs, and for richness and evenness, so the index will combine both of the concepts. The most used index are Simpson diversity and Shannon index. Again, I will give you a very simple example just to understand the mindset of the alpha diversity, and then we'll show you a practical result you can see in a publication, or we can present to you in our research. So here you have two samples with the community 1 and the community 2. A and B, in both cases, you have 5 species, one blue, one orange, one green, one yellow, one red. So it is exactly the same richness. But in the case of the sample B, you can see that the blue species is dominant. And so it means that B is less evenly distributed than A. And then we will conclude that the alpha diversity, when we talk about richness plus evenness, is higher in A than in B, mainly because of the evenness, because if you look at the number of species, this is the same. And in fact, we will have the confirmation of the richness of the alpha diversity with the rarefaction curves. Practical example, again, coming from the same publication, you will have here the number of OTU, so what we call the richness. And here on the right, you have the Shannon index, what we call the alpha diversity, defined as evenness plus richness. And again, on the left, you have the lumen part of the microbiota, and on the right, the mucosa for ileum, cecum, and colon. And just as a quick example, you can see that for the microbiota in the lumen of the ileum, the number of OTUs is lower than the one for the colon, and you will see the same for the Shannon index. For the beta diversity now, the third key parameters, the definition is the difference or the similarity level between different communities and samples. Here, the objective is to understand how similar or different are microbiota profile of two or a group of samples. And here, we will use multivariate analysis, also called PCOA or NMDS, which are based on ordination methods. Again, as a simple illustration, on the right, you have group 1, and on the right, on the left, sorry, and on the right you have the group 2, and you will see that in the first group, the composition of the microbiota of the 3 pigs is very similar among samples with the same color, and you see that the microbiota composition is very similar along all samples, and then you will have a low beta diversity. On the contrary, on the group 2, you can see that from one individual to another one, the microbiota composition is different, and it means that you will have high beta diversity. After that, you will calculate the distance, and you will be able to see how different are your communities. If we go a little bit more into details about the ordination technique, what it is a statistical method that uses a set of multiple variables to distinguish samples from each other. And we have dedicated software using algorithms, and they will determine the best cluster of OTUs to discriminate samples. And after that, the microbiota sample will be represented as a point. This is important within 2 or 3 dimensional space. And after that, you will be able to calculate the difference by specific tests like Permanova or Break-Restis distance. Basically, when you have a short distance, it means that the microbiota composition is very similar, while when you have a long distance, the composition and the structure of the microbiota is very different. And again, if we have a look on the publication, this is the result the author obtained. And you can see here, it's the author plots the different microbiota between ileum, cecum, and colon. And on the left, this is for the lumen, and on the right for the mucosa. And you can see that the beta diversity here, so the structure of the microbiota is different for ileum than for cecum and colon, while for cecum and colon, this is the same plot. And if we look at the mucosa part, you will see that you have 3 different structure community depending on the intestinal tract location. So what are the key points for the amplicon sequencing? The first one, that it is based on gene amplification and on gene marker methods. Amplicon sequencing allows to study microbial community composition, microbial network, and it is a tool to understand microbiota with 3 main components. The first one is the percentage of relative abundance. So it is a proportion of a given microbial species in a microbiota sample. And you will be allowed to do differential analysis to see how different is your microbiota from one sample to another one. You have after that the alpha diversity. It is the diversity within one microbiota sample. And it is defined as the number of different species present, so the richness, and the relative abundance, the evenness. In other words, the dominance, if there are some bacteria that are dominant compared to other one. And finally, the beta diversity, it deals with the microbiota structure, and it is to evaluate the similarity between different microbiota samples. And then here, to evaluate this beta diversity, you will use multivariate approach. And once we have these key parameters, we can go further to understand the microbiota structure and the impact on the host. And then I just wanted to give you 2 examples of what we can do to go further. And the first one is to describe the microbiota structure. And it is when I want to classify my samples into known groups and to identify the key OTUs, allowing this discrimination. So you will use here discriminant analysis like the LSDA, or the linear discriminant analysis effect size, the LEFSE analysis. And I put here an example of the Lefse analysis. And so I want to know how my population is characterized. And this population is comprised in two different classes, like for example, diets, two different diets. And in this case, it's dysentery inoculation. So you will have some piglets that will be positive to the dysentery in green, and piglets that will be negative to the dysentery in red. And when you do the Lefse analysis, you will be able to tag what are the bacteria that characterize the group 1 and the group 2. So here, for example, this taxa will be more abundant and characterize the group with dysentery, while the other one here will be more abundant and will characterize the group without dysentery. The LDA score you have here is a calculation of the discriminant power you can have with these different OTUs. The second practical analysis example I wanted to give you is how to link the host rate to the microbiota feature. And this is actually a paper that we have just submitted to one scientific journal. And here in this trial, we use fattening pigs, and fattening pigs were subjected to 3 different periods of heat stress. The P1, so the period 1, was the first period of thermoneutrality, and P2 and P3 were second and third period of heat stress. And we wanted to see how the microbiota can impact zootechnical performance and also the converse. And what you can see, so we use regularized canonical correlation analysis. It is a multivariate analysis that allows maximizing the correlation between 2 data sets. And here, in this case, the microbiota dataset and the zootechnical parameters. The zootechnical parameters are here. So, for example, DMI is for dry matter intake during P1, and RE and ME ratio is the ratio of retained energy on metabolized energy for P3 or P2. And what you can see is that here is that we selected some OTUs from the P1 microbiota dataset, and we obtained, for example, and we obtained, for example, this OTUs, Ruminococcus probably bromii species, which was positively correlated with dry matter and retained energy during the heat stress period. This is what you can see here with the red color, which is the correlation coefficient. And so the hypothesis we made is that the composition of the microbiota at the end of the first period, so during the thermoneutrality period, can influence energy balance and dry matter intake during the heat stress period. This is what you can see when you look at the correlation between these OTUs and the return energy or the dry matter intake here. And on the converse, we also have a look on the dry matter intake in P1, and you can see that the level on intake in P1, so again during the thermoneutrality, is closely correlated with some OTUs in P3 in a negative or positive way. And the second hypothesis we did is that the level of ingestion before a stress period can shape the microbiota during the heat stress period. So, conclusion on perspective toward a holistic understanding of the microbiota using amplicon sequencing and other techniques. Today, we made a focus on amplicon sequencing, and you saw that it allows us to understand the community composition and to do some microbial networks, and also to make a relation with the zootechnical parameters. But of course, now we want to go further and to go from the microbial potential to the microbial function. And we work also on different techniques, the final objective being to have an holistic view and to understand what it happens in the swine. And we strongly believe that working on different techniques, from the 16S rRNA to omics technology, will open us avenues to understand not only microbiota, and so the composition of the structure, but also the function, microbiome, and their implication to enhance health and productivity in the farm, to increase the sustainability of our livestock system, and as a producer of probiotic, of course, to draw some precision probiotics and to decipher in a very detailed way the microbial mode of action. And we strongly believe that doing that will allow us to deserve personalized farming. As a conclusion, because it is very important for me and for us, we go toward a more holistic approach, and we know now that everything can be correlated. And then to use all this method in a successful mode, we really need to discern hypotheses and concepts and to have a clear idea on what we want to achieve. So I thank you for your attention. I hope that this presentation is useful for you to understand all the results we can have around the microbiota structure. And I will let the micro to David, who will show you how we can use the 16S amplicon sequencing to objectivize the effect of our probiotics in swine.
Thank you very much, Emmanuel. Yes, I'm here. I hope that you hear me. Do you hear me, Emmanuel?
Yes, sorry, yeah.
Okay, perfect. All right. So, Emmanuel, please go to the next slide. And well, here, after this wonderful presentation from Emmanuel, I would like to show you an example developed with our probiotic with Levus LSB specific live yeast in swine, so that you can see the kind of results that we can obtain with these methods, with the 16S rRNA amplicon sequencing. In this case, I am going to show you a trial done with a large integration. In this case, we supplemented the sows or not with the probiotic Saccharomyces cerevisiae variety boulardii. The sows were supplemented since 4 weeks before farrowing and during the lactation period. And then the piglets were receiving different diets, either a controlled diet or a medicated diet. We took fecal samples from the sows and also from the piglets. From the sows, we took samples 5 days before farrowing, then 24 hours after farrowing, then on the day of weaning, 6 days after weaning, and 20 days post-weaning. I would also like to mention that this study was published by our colleague Caroline Achard last year at the International Scientific Conference for Probiotics, Prebiotics, and Gut Microbiota and Health in the Czech Republic. So if you can go to the next slide, Emmanuel, please. So here, the first results that we saw, we analyzed the alpha diversity. If you follow the gray dots, the gray color dots, this is the control sows, and we analyze the Shannon index, which is one of the parameters that Emmanuel has been presenting before. Having a look to the control sows, if we compare the sows 5 days before farrowing and 24 hours after farrowing, what we can see is that the alpha diversity is being reduced during the farrowing time. So the sow is some way losing heterogeneity in the microbial population in the gut. Also, what we can see is that the samples in the control group 24 hours after farrowing, those samples are very heterogeneous. I think that I made a little bit of a mess. The alpha diversity is coming down in the 24 hours after farrowing, but when we compare this alpha diversity from one sow to the other sows in the group, what we can see is that the range of alpha diversity is huge. Then when we are applying the probiotic, the Boulardii strain, what we can see is that the sow's microbiota, at least from an alpha diversity point of view, seems to be much more stable compared to the sows in the control group. And this is quite important in sows at farrowing. Next slide, please. And here, when we analyze the relative abundance of the different groups, what we can see, again, one of the conclusions from this slide is that sows fed with Lewis LSV, it looks like the microbial profile is more stable compared to the sows in the control group. If we see as an example Firmicutes, Firmicutes is a group, the relative abundance of the Firmicutes in the sow feces is usually quite high, but what we can see is that in the control group of sows, this Firmicutes group is reduced by more or less 10%, comparing 5 days before farrowing and 24 hours after farrowing. When we are applying the probiotic to the sows, we do not see such a reduction in the level of Firmicutes. Maybe more important or more interesting for us, for swine nutritionists, is what is happening with the proteobacteria. Proteobacteria is a big phylum of bacteria containing Escherichia coli, Salmonella, Shigella, etc., Campylobacter, etc. So it is a group in which I would say contains a big portion of the typical pathogens that we can find in swine nutrition. Usually this is important also because in sows around farrowing, it is very typical to see an overmultiplication of proteobacteria. And the impact that they may have is that these proteobacteria can be transmitted to piglets at birth, because at the end of the day, the piglets are going to get contaminated from the first, let's say, contact with the feces of the mother, with the skin of the mother, etc. What we can see here in this graph is that the proteobacteria are over multiplying quite dramatically in the control group of sows, while in the group of sows in which we are applying the probiotic, the proteobacteria are also multiplying, but what we can see is that they are not multiplying at the same level as in the control group of sows. So some way we are helping the sow to produce a more healthy microbial profile to the piglets. And finally, when we have a look to the Fibrobacteres, as you can imagine by the name, is a group of bacteria which are able to ferment fiber, to use the fiber. And what you can see when we compare the microbial profile, the level of Fibrobacteres in the control sows, it is much smaller. The relative abundance in the control sows is much smaller compared to the sows fed with the Boulardii strain. Actually, this is part of the mode of action of this specific probiotic. This is important because the more bacteria able to ferment fiber you have, the more energy you can extract from that fiber fermentation. And this is happening before farrowing. As you can see, after farrowing, the level is very similar and very low in both cases. The hypothesis here is because, first of all, we are already changing the diet of the sows, so the amount of fiber is lower in sows entering into the lactation period, and also the feed intake, so probably the total amount of fiber reaching the gut, the hindgut of the sows, is also quite low, and this is why there is a lower level of Fibrobacteres in those groups. But overall, as I was mentioning, what we can see is a more stable microbiota in the sows fed with the probiotic. Next slide, please. And finally here, using this ordination technique that Emmanuel has been explaining, when we were taking samples of the piglets 20 days after weaning, the piglets were not receiving the probiotics, so they were receiving different diets. And as you can imagine, the effect of the diet is usually the most important one to describe the microbiota of the piglets. Even 20 days after weaning, with the application of this ordination technique, with the application of amplicon sequencing, we could see how the microbial profile of the piglets coming from sows fed the probiotic was different from the microbial profile of the piglets coming from control sows. This is, I would say, neutral information. Only with this graph we cannot say if this microbial profile is better or worse. I mean, we were lucky in this trial because the performance results of the piglets coming from sows fed with the probiotic was much better than the performance results of the piglets coming from the control sows. And then something else that we detected in the piglets after weaning, at weaning, 6 days after weaning and 20 days after weaning, it was that we were able to detect a higher, the detection frequency of Fibrobacteres was higher in the piglets coming from sows receiving Lebus LSB, receiving the probiotic. And here it is an illustration of the maternal microbial imprinting, which is a concept that describes how feeding the sow with some specific nutrition, with some specific ingredient, we can still see an effect on piglets, but quite late. I mean, even 20 days after they have been separated from the sow. Next slide, please. And I think it's already the last one. So just to conclude, to take some take-home messages from this trial, farrowing in sows represents a critical period. It is associated with hormonal changes, with stress, and also with alterations of the microbial profile, which typically are shown on the form of constipation. Is very typical in sows around farrowing. The microbial profile, as we have seen, can be stabilized with the application of specific probiotics, also to drive a better microbial transmission from the sow to the newborn piglets. And something else, and I think that this is quite important, the effect of sow nutrition goes far beyond weaning. So when we are working with sow nutrition, we are not just improving how the piglets are going to be at weaning, but also how those piglets will develop also after weaning. It will have an effect, as we have seen, on the piglet microbial profile, and something that we can see that we have seen is that piglets fed with the same diet after weaning can show different microbial profiles depending on the diet that they received from the mother. What are the applications of this? Well, on one hand, we can think about this to improve the piglet performance in the nursery, and also that would be a very useful tool to apply in the medication programs. I mean, when we talk about the medication, we tend to think about working on piglet nutrition only, but the best start for the medication program is really starting with the mother. So that was everything from my side, from my presentation, and now we have some time for a few questions from your side. Please feel free to write your questions in the panel that you have on the right-hand side of your screen, and with Emmanuel we will try to answer those questions. The first one I would like to ask you, Emmanuel, is, I mean, while we can see if some more questions are arriving, Emmanuel, we hear more and more about the gut-brain axis. Could you tell us something about this concept and what is its importance in swine production or animal production?
Yes. Yeah, you're right, David. It is more and more recognized, in fact, that the microbiota can impact the behavior of the animals, including the swine. And more and more articles speak about a bidirectional dialogue between the host and the microbe. And there are several neurochemicals that act as a let's say, interkingdom-based language between the microbiota and the host. And the few examples can be catecholamines that are produced by the host, but we know that also microbiota can. Short-chain fatty acids and probably other molecules that are known to act. And especially for the swine industry, I can see already very nice perspective. Like the management of the sow behavior around the parturition. As you mentioned, it is really a stressful period. And probably the modulation on first the understanding of the role of the gut microbiota and then the modulation of it could be a way to investigate the gut-brain axis in order to decrease the stress. Another very nice potential application could be to limit the anorexia during the weaning. And another one could be, for example, to impact feeding behavior during stress period, like heat stress. And basically, this is something we saw in the publication which has been submitted, and I show you a few results with the correlation between the technical parameters and the OTUs. And we suspect that feeding behavior can change can be changed, sorry, with the LSB, with the Levus-LSB. And then the feeding behavior is probably linked to the gut-brain axis. So this topic is really interesting, and for all kinds of animals, we can see a lot of perspectives of research.
Okay, thank you very much, Emmanuel. I have now a question. Maybe this is very broad, I don't know. How do the ingredients each affect to gut microbiota? I mean, I don't know if you have some idea about how the application of different ingredients into the feed. I think that here it is more the difference is when you are feeding a fiber ingredient or a milk ingredient, etc., would have an effect on microbiota.
Sure, the ingredient can impact the microbiota in a strong way. We already know that the high fiber diet will impact the community, as well as high starch diet, but also lipids. Everything can impact, in fact, the microbiota. This is the difficulty, but also the beauty of this research. You have to take into account the feed ingredients when you try to decipher the effect even of our probiotics. We try to also analyze the diet, just to put in perspective the impact of our probiotic and the role that could have played the diet given in a given trial. I hope it is clear.
I think so, Emmanuel. Thank you very much. Another question. Do we also take into consideration the, I mean, sorry, I am reading only part of it. When comparing relative abundance, how do we also take into consideration the total number of microbes present?
This is a good point. Actually, there are more and more discussion around the normalization, because most of the times we do not have the total quantity of bacteria, and there are several methods of normalization, and we can start to see it in different publications, and this is something we are working on as well, just to be able to have not only the relative abundance, but also the 100%. What does it mean? And this is something that was not really implemented in a huge way up to date, but we see that it's a practice which increase, and we are working on that as well.
Okay, thank you, Emmanuel. I think that I will launch 2 more questions, and then we will finish the webinar. So the next question is, then could it be possible to, I mean, it is related with the answer you gave about gut-brain axis. The question is, then could it be possible to measure the microbial signals in the gut which could be responsible for gut-brain axis dialogue?
Yes, typically the metabolomics method could be used to do that, and you can either choose to have non-targeted metabolomics. It means that you will go without a priori on the molecules you want to obtain, or you can do a priori, and then you will do targeted metabolomics to target specific metabolites that you want to measure, to quantify in a specific context, and to see what is the role of this specific molecule.
Okay, thank you, Emmanuel. Last question, but before launching the last question, I would like to advise the people, I mean the attendees, when you are leaving the webinar, there will be a survey appearing in your computer. Please take 2 minutes to answer this survey, because it will help us to also to improve the organization of these webinars. It is going to be quite fast. So the last question is, could non-starch polysaccharide enzymes have effect on microbiota? How to evaluate the right or bad impact on it?
Non-starch polysaccharide enzyme.
Yeah, like silanase, beta-glucanase, etc. I mean the yeah.
So you mean the yes? Can you re-ask the question, please? Repeat the question.
Yes. Could non-starch polysaccharide enzymes have effect on microbiota? How to evaluate the right or bad impact on it? I think that the question is asked from the perspective that some of the ingredients, especially, well, or some of the components included on the cereals, like silans, etc. This portion,
Yeah.
I mean, some of them, they are going to be modified with the application of non-starch polysaccharides.
Yeah.
And what would be the effect of this addition, of this modification, on the microbial profile that we could get?
Okay, so it means the addition of enzyme in the feed.
In the feed, yes, yeah.
Okay.
I mean, I
okay,
understand that it goes that way, yeah.
Yes, probably yes, because you will pre-digest on principle. This kind of enzyme are produced by the bacteria, and they if we speak about fiber, the fiber will be digested or degraded by the bacteria who has this kind of enzyme, and will produce short-chain fatty acids. But depending on the enzyme activity, and I am not a specialist of the enzyme added in the diet, but we can imagine that you will pre-digest, and then you will have shorter oligosaccharide, and then it will be a different pattern of digestion in the microbiota. But honestly, it requires some tests to evaluate the impact.
Okay. All right, Emmanuel, so we are at the end of the webinar. Thank you very much for your presentation. Thank you also to the attendees for your questions and for your participation at the webinar. It's a pity that we cannot see each other, and we are forced to do this kind of events in a virtual mode. But anyway, I hope that you found it useful, that you learned about metagenomic analysis, about amplicon sequencing, about how to study the microbiota, and therefore this information has also opened new ideas, new research ideas, etc., so that you can apply them in your day-to-day work. So thank you very much, and have everybody a very nice rest of the day. Bye-bye.
Thank you. Bye.
Thank you, Manuel.